Hello, my name is Mr. Chippen, and I am the AP Biology teacher at Murray High School in Murray, Kentucky. And this is Unit 4 Cell Communication Cell Cycle Review. We have an exam coming up. Hopefully, you are preparing. Hopefully, this video is helping in your preparation. If you haven't watched my other videos, you should do so. They can also be helpful to you. If you like this video, then hit the like button. If you would like to tell me that you like it, please comment down below. Also, subscribe to my channel for future content because I'm putting out things pretty regularly. Let's go through and talk about cell communication. In this video and the next video, we'll talk about cell cycle. Cell communication. It's when cells communicate with one another. How do they do so? Well, they do so sometimes by touching, like literally directly touching. Direct contact is what this is called. There's several ways that this can occur. We talked about plasmodesmata found in plant cells. In animal cells, there can be these gaps called gap junctions. They're literally what they're called. And so they allow for direct contact with cells. Another example of this is immune system activation. So you have a cell that presents an antigen. Antigen's basically like a little protein that says this is the thing that I just killed. See, it's showing up that antigen. The helper T cell is like, let's do something about this. And then it's going to go and recruit the soldiers. Those soldiers are B cells. And it's going to present that antigen to the B cell. See, they are having direct contact with one another. It's not affecting these cells over here. It's not affecting the cells that are in your feet or wherever else may be. It's only affecting that cell. And so that is an example of local, or not local, direct contact. Moving on. There's distance contact as well. I love Shutterstock images. Look at it. It's perfect. So distance communication could be like a local thing, like me communicating to my classroom, or it could be a distance thing, like me communicating to you. I don't know where you are, but you're probably not in the same room as me. And so it is a distance thing. Or you could think of it as me communicating to my classroom, be local, and long distance would be like the office communicating with the whole building via the intercom. That may be another way of thinking about it. Let's talk about examples of each. Before we talk about examples of each, either one of these have a target cell. Target cell is the cell that is designed for the message, meaning that that message that is being sent has a target. When someone calls my classroom and says so-and-so is being checked out and needs to come to the office, that message has a target. No one else in the room has that name, and so that message has a target. Those target cells will usually have some sort of receptor on them that makes them the target. Notice this is not a target cell because it doesn't recognize these purple diamonds. It doesn't recognize them because it doesn't have a piece that recognizes them. This cell does have a purple diamond piece, and so that makes it a target cell. So let's talk about in the local type of communication. This would be like, again, me communicating to the classroom. Maybe a cell is secreting some business that has target cells in the vicinity. So let's say that there's an injury, like maybe you get stung by a bee, and those injured cells are going to release a local regulator like histamine in order to communicate with the regional cells to initiate like an inflammatory response. So that would be an example of that. This is a nerve cell releasing neurotransmitter that is dealing with these cells, or this particular cell in this region, but any cell could pick up that neurotransmitter and be stimulated by it. Long distance has to do with when one part of the body is releasing a chemical that affects a different part of the body. Usually in the endocrine system. In the endocrine system, hormones are being released directly into the bloodstream. And so this would be an example of long distance. Still, we have that concept of the signal molecule being created by the cell and a particular target cell. I think I have some examples of those. Neurotransmitters gave that example for the regional example. Long distance. Let me talk about an example. So you have a cell, let's say that you have a cell in your parathyroid gland. Parathyroid produces a hormone that regulates calcium levels in the body. What is it going to talk to? Well, it talks to the bones because that's where that mineral is stored. And so it's going to release a hormone, and the only cells that are able to recognize that hormone are bone cells. No other cells are able to recognize that hormone. That would be an example of long distance. Parathyroid's in the neck, bones are in the bones. So let's talk about the signal transduction pathway. Signal transduction pathway is a pathway in cellular communication that has three parts. This picture I'm sure that you've seen in your own classroom. reception, transduction, response is the three portions of this. The reason I have a picture of a phone here is because, well, when you look at your phone, it's ringing. This is an old phone. Look at that 3G, just proudly showing itself. This represents reception here, right? This is reception, meaning that you see the response, or you see the thing that you should respond to, and you see that green button that says answer, and that is telling you answer the phone, or just simply me hearing the phone ringing in my classroom, and that is me receiving that response. Transduction has to do with all the pathways between reception and response. Usually in a cell, it's going to be, there we go. In a cell, it's going to be a series of proteins. That's what this is showing. You have these different kind of proteins that are changing shape in order to do a thing. And so that's going to be any kind of, basically like a relay pathway is the way to think about it. So like when I answer my phone, when I answer my phone, or when I hear my phone ring, sorry, I hear it, I walk across the room, I pick up the phone, I listen, I receive a message, and that message is so-and-so is being checked out. All right, all of that would be the transduction of that particular pathway. The response is me saying to the student, you are being checked out, and then that student getting up and walking out of the room. So let's talk a little bit about these three steps. Reception. So reception has two parts, a receptor, which makes sense, and a chemical messenger, which is generically called a ligand. So the ligand or a ligand is any kind of chemical messenger. There's the ligand binding domain. This is basically the receptor. Notice that this binding domain has a particular shape, and that shape determines its function. Very much what we are used to seeing at this particular instance, right? That this shape that looks like the symbol for the post office has a shape, and it's not, nothing else is going to be able to bind to that particular ligand. The receptors can be located inside or out of the cell, depending on the type of hormone or the type of hormone that it's receiving. If it's a steroid hormone, remember steroids are lipids, and that's going to be received inside the cell. Why is that? Because steroids can pass directly through the cell membrane because they're lipids, and they can pass through the cell membrane, so the receptor is going to be found inside. You still have the transduction, you still have the response. I didn't say this. Response is usually some sort of protein is going to be made, right? Because proteins are how things get done in the cell, and where are the instructions to make the proteins on the DNA. So there's going to be some sort of, that's what this is showing you here, DNA, messenger RNA, protein synthesis is happening, transcription, transcription, translation, you're familiar with those things, I'm sure, by this point. And if it's a non-steroid hormone, it has to be received on the outside, because anything that has any kind of polarity cannot pass through the cell membrane, has to be received by a receptor protein, initiates some sort of cascade of responses, and then you have the product that is being made, usually a protein. Speaking of cascade of responses, here's a signal cascade. I like to think of the signal cascade as kind of like an old-timey phone tree. Back in the days when they didn't have like all calls and those sorts of things, you were responsible for calling two people. Like if we were going to be out of school, you called two people and you told them there's no school today. Well, those two people would call two people each. And so my two calls have now been responsible for 4 calls. And then those 4 people are going to call 2 each. And so now what is it? 14 people have been by my one phone call, right? Or the one phone call that I received. 14 other people, 15 counting myself, have received this message. And if you keep going down, that's going to cascade. And so what this is showing you, that one single chemical can cause a cascade of responses. And this is just to reserve energy for the cell. This is called signal amplifications, another way of thinking about that. You may see that in the literature as well. And protein modification, this just has to do with the proteins being changed. You know, we've talked about the protein shape determines its function. Usually that change has to do with phosphorylation. When a phosphate is added, phosphates are highly negative, it's going to cause the shape of that protein to change, and that's going to determine and change its function. Pretty straightforward. You see this in a lot of signaling pathways, where this shape is going to change based on phosphates being added to it, and it's going to cause it to do something else. Like this, for instance, secondary messengers. So here's the reception right here. This little candy corn is going to be received on this thing, and it's going to cause this protein to change shape, which is going to bump into this protein. G protein is a pretty common messenger on the cell membrane. That's what this GTP is, like just another like ATP sort of thing. And it's going to be phosphorylated, cause it to run over here and turn on adenylyl cyclase, and that's going to cause secondary messengers. Secondary messengers is a fancy way of saying just a relay molecule. CAMP is an important secondary messenger. CAMP stands for cyclic AMP. It's just like ATP and ADP, except for it's adenosine monophosphate, but it's just cyclic. Environmental influence. Let's talk about environmental influence. The environment can influence cellular response. Pretty simple. I have an epinephrine response here because if you're walking in town, you round the corner and you see 30 dogs all snarling at you, your body's going to be like, hey, the environment's changed. We should have a particular response for this. Your body's going to produce a chemical called epinephrine, and that's going to cause a cascade of responses. It's going to cause lots of things to happen. Glycogen's going to be used in order to give you extra energy. Blood vessels are going to dilate. Chemicals, all this stuff is going to be happening to cause you to be able to run a little bit faster, not faster than those dogs, so hopefully that doesn't happen to you. And this is going to cause change in gene expression. And so this is important. Environmental responses cause a change in gene expression. What do we mean? Not these things, these proteins aren't normally being made, right? And so when you receive that, it's going to cause you to start making things that you otherwise weren't already making. I think that's just pretty self-explanatory, but we're going to say it anyway. Apoptosis is an example of a cellular response that is a response to the environment. An individual begins developing at a particular rate, and that development takes place, the development of a hand, for instance. Most people, their fingers are separated, thankfully. And how do we get rid of those webs in between our fingers? It's apoptosis. How do those things know to disappear? There's some sort of environmental cue that says it's time for the fingers to be separated, and then they do. If that environmental cue doesn't happen, then a baby could be born with webbed hands. In bacteria, this is called quorum sensing. Quorum sensing is the idea that bacteria can sense when other bacteria are around and elicit a certain kind of response to that. One of the responses that's often associated with quorum sensing in bacteria is the production of something like a biofilm, like just this it looks gross, nasty kind of film over the surface of like liquids and things, and they produce this when there's enough bacteria around. It just senses quorum just means like, is there an appropriate number of people or of bacteria in this case, and sensing means that they're able to sense one another's presence. What else could cause a change in the response? Mutations. Change in the DNA, change in the protein, change in the response. This is showing UV photons could change that. There's lots of things that can cause mutations. This is going to cause a change in the signal transduction pathway. It's not going to, the secondary messengers aren't going to do, the nucleus is never going to say, hey, what are we supposed to do? They don't know because they never got the message. Pretty straightforward, common sense kind of thing. Another one is another example. This is a chemical blocker, something that's blocking the actual reception or the release of that chemical could also change the signal transduction pathway. Carbon monoxide is a great example of this. Not a great example because we don't like it. It's not good. But, oxygen is the final electron acceptor in cellular respiration. Well, carbon monoxide comes and binds to the place where oxygen would normally do its thing, and thus blocking oxygen from doing that, causing the individual to not be able to get oxygen, which is not good. And so this is an example of how those those pathways can break down under certain circumstances. Hopefully this was good. Hopefully you enjoyed this video. If you did, watch the next one. Watch the previous ones. Watch them again. Subscribe to my channel. Please help me out. Share that knowledge of my channel with others if you think they could help them. Thanks for watching.